Hello, everyone. Welcome to today's live broadcast, the use of a CD200R inhibitor to overcome central nervous system tumor-induced immunosuppression. I am Brenda Kelly Kim of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by Immunochemistry Technologies. Immunochemistry Technologies provides quality laboratory service and research reagents. As a contract service laboratory, ICT specializes in custom immunoassay development, manufacturing, conjugation, lyophilization, and consulting services. ICT is focused on ELISA development and has the knowledge to develop reliable, sensitive, and specific tests. ICT offers a line of ELISA diluents, including coat buffers, conjugate stabilizers, and sample diluents designed to reduce backgrounds, increase the specific signal, and improve the stability and shelf life of the ELISA. ICT also offers a line of fluorescent reagents to measure capsase activity, apoptosis, and cell death in vitro. For more information, please visit www.immunochemistry.com. We have a few announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit your questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to get to as many of your questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window to let us know, or submit your problem through the green Q&A button lower left. This presentation has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. I would now like to introduce today's speaker, Dr. Michael Olin. Dr. Michael Olin is an assistant professor in the Department of Pediatrics at the University of Minnesota. His research is primarily dedicated to developing immunotherapy for brain tumors. He utilizes tumor cells as vaccine components, demonstrating promising results with minimal toxicity. Dr. Olin received his PhD in 2005 from the University of Minnesota's Veterinary Medicine program. I will now turn it over to Michael for his presentation.
Well, thank you very much. Please bear with me. I have a little bit of a Minnesota cold, so I'll try not to cough and make anyone sick over the internet. My lab focuses mainly on immunotherapy, immunology, and ways we can exploit the immune system to overcome tumor-induced immune suppression. And we've been doing this with some success over the last few years. So basically, the progression to a productive immune response involves passing a number of what's called immunological checkpoints. And these checkpoints are called checkpoint blockades. In other words, when they recognize tumor cells or light antigens from the tumor, like we give as the vaccines, they can actually shut down the immune system and inhibiting the ability of us to mount an anti-tumoral response. So people have been looking at ways to overcome this induction of immune tolerance through what's called checkpoint blockades. And one of the more famous ones right now is an anti-CTLA-4, or looking at the CTLA-4, see if I can remember how to do this, blockade here, which shuts down a T cell activation. Another one is called through programmed death ligand 1 and its receptor PD-1. In other words, the tumor cell can actually exploit this in the tumor microenvironment, virtually shutting down or inactivating T cells. So people have been looking at this with some success by deriving antibodies, humanized antibodies, against the CTLA-4 and the PD-1, inhibiting these reactions and eliciting an immune response. And they've been doing this with some success, specifically in melanoma. Well, we're chasing down a third immunological checkpoint called CD200. CD200 and its interaction on the CD200 receptor induce antigen-presented cell tolerance, inhibiting the ability to make an immune response or elicit an immune response. So basically what CD200 is this extremely immunosuppressive protein. It negatively regulates the immune system, or it shuts it down. A lot of preliminary studies on graft rejection show that if you give mouse CD200 protein, it'll never reject the graft. It shuts down the immune system totally. And that's through binding on its receptors, which are mostly on myeloid cells. However, following TCR activation, the CD200 receptor is upregulated on T cells, which we'll get into that problem in a little bit later in this talk. So, like I said earlier, tumors induce suppression. There's multiple mechanisms, there's multiple ways that the tumor is capable of this. And one of them is through antigen presentation. Now, Gorzinski et al. Reported that the CD200 and its receptor interactions induces tolerogenic DCs. And this really sparked our interest because we want to inhibit this induction of tolerogenic DCs to elicit an immune response. Moreover, it's been suggested that CD200 expands what's called a myeloid-derived suppressor cell population, which induces immune suppression. I'll be showing you some data that we've done in-house and I've actually published showing that this is indeed true. Now, in the central nervous system, neurons highly regulate the CD200, and that's to inhibit activation of T cells that you don't want in the central nervous system. And it's been reported that downregulation or the loss of the CD200 is correlated with increases in MS as well as Alzheimer's. It's also been shown that CD200 is expressed on multiple tumors, including melanoma, pancreatic, breast cancer, and what we're interested is glioblastoma multiforme, or a CNS disaster. So we started looking in on healthy donated CNSs at autopsy, and we showed that CD200 is highly expressed, at least by transcription, throughout most of the central nervous system. And you can see here, in brown, on the brown, that's protein levels in the normal central nervous system of CD200. Now, this isn't true in peripheral organs, such as breast, kidney, liver, pancreas, and lungs. You can see that there's relatively low expression of CD200. So we went on and showed that through multiple CNS tumors, such as meningiomas, glioblastoma multiforme, medulloblastomas, et cetera, you get high transcription. Well, compared to meningiomas, which isn't a typical CNS tumor, you get high transcription of the CD200 protein. So we decided to look at the potential use of this in immunotherapy. Because the CD200 receptor interactions have been demonstrated, although there's some controversy over this, that they enhance metastasis of breast tumors and multiple myeloma. And it's been reported that blocking the CD200 receptor actually has shown to be decrease tumor growth. Although, like I said, there's a little bit of controversy over that, and we're trying to figure it out right now. So there has been clinical trials using an anti-CD200 where this company targets the CD200 on the tumor on the tumor itself in B-cell chronic lymphocytic leukemia and multiple myeloma. However, there's not much talked about, or there's no follow-up on the outcomes of this program. So we think this is because what we discovered just recently, that you can see the brown in normal autopsy tissues, CD200 staining in brown. You see very little of this on the tumor itself, especially high-grade glioblastoma multiformes. And that's due to the protein being cleaved by a ADAM28 protease. So we think that targeting the tumor itself, the CD200 on the tumor, will not be efficacious because it's solubilized. And that correlates with some patient data, including ours, where Wang et al. Showed or reported that it's a soluble CD200 that regulates or correlates with poor patient outcome. And we reported this earlier this year, where we took patient samples from an ongoing or a finished university clinical trial of ours on gliomas. And I also had serum samples from some friends, colleagues of mine, where we showed that in glioblastoma multiforme, there's some high levels, some patients had high levels of CD200 in their sera. Now, what was interesting are these patients correlated with real aggressive active disease. If you look further on this graph, this medulloblastoma was a patient of our clinical trial that we just reported. And this was at the point where she actually went off trial because she had rapid progression of her disease. But what was very interesting and caught our attention was within all these ependymomas, one of the ependymoma patients was on our trial. And when she came in, she had relatively low concentrations of CD200. And she actually responded to our therapy, which was pretty exciting. But what happened was when she had reoccurrence at week 8, her CD200 level significantly increased from her baseline. And then when she had her spinal mets, it even went up further. Moreover, you can see that this increase in soluble CD200 correlated with an increase of her myeloid-derived suppressor cell population as she went off trial. So when her tumor came back, it solubilized the CD200, this is what we're hypothesizing, and it increased her MDSC levels as she progressed, her disease progressed. And this was true with our glioblastoma multiforme patients as well. So we asked the question, how can we block CD200 induced immune suppression and MDSC expansion? Well, what it turns out is that this is our hypothesis right now. This is our working hypothesis in my lab. There are some holes in the story because it's ongoing research. But what we've shown, or what's out in the literature actually, is there's a CD200R1, receptor 1, and there's isoforms, which are termed R2, R3, and R4. Well, it turns out, at least in the literature, and we're testing this right now, that the CD200 protein itself, which is extremely immunosuppressive, binds to the CD200 receptor and delivers an inhibitory signal through a DOCK pathway, DOCK1 and 2. These do not bind to CD200 receptor 2, 3, or 4, or the isoforms. At least this is what's told in the literature today. So Gorzinski reported that if you took peripheral blood from 2 donors and added them together and induced what's called a mixed leukocyte reaction, you could induce a cytolytic response. He's also measured this by interferon gamma. Excuse me. And he showed that if you add the protein, the CD200, you can inhibit the production of a cytolytic response, which has been shown it's an immunosuppressive. But what he started doing is he looked at crystallography, and he actually looked at regions of the CD200 protein itself, which when you add it to the mixture, you either got this agonist, which he's terming as an agonist response, or an inhibitory response, the same as the protein. But what was interesting is different regions of the protein, you got an antagonist response, or it overcame the suppressive effects. So we started looking at these, and what we're hypothesizing, and this is a hypothesis based off one of Gorzinski's literature or manuscripts as well, is that potentially these peptides bind to the isoforms, and these isoforms actually activate, that didn't turn out well, activate an immune response. So to me that, well, we're kind of going back on this antagonist versus agonist right now. But for this presentation right now, until we totally characterize it, we are going to call these CD200 receptor antagonists. So that's the terminology I'm going to use for the rest of the talk. So we decided to test these antagonists, and we went back and actually derived different peptides based off crystallography. But what we showed is that if we give a mouse a glioma and we vaccinated with only the peptides, that CD200 receptor antagonist on day 2, and I'll talk about why we had to go a day early with the antagonist a little bit later, but then we vaccinated with OVA plus poly-IC plus the peptides or the antagonists, we could show that in non-tumor bearing mice, we could actually, using OVA, elicit an immune response. We can get this significant expansion of a CD8 positive, syncytial positive T cell population. If you just use tumor bearing mice and give it OVA, you lose that ability to expand. You get an immunosuppressive effects in the draining lymph nodes or the cervical lymph nodes of the mouse. However, if you give it our antagonist, it should be antagonist, I apologize, you get a complete reversal, or you get this expansion. And we also showed that functionally you can overcome the immune suppression, the immunosuppressive effect of what we're thinking is soluble CD200. Now, we went ahead with the help of Immunodex. We got some detrimer with Dr. Haley, and we showed that if we stain with these detrimers that are specific for TCRs that are syncytial positive or reconfigured for syncytial, you can see that there's a nice TCR expansion in the draining lymph nodes of non-tumor bearing animals. We lose that if we look at the tumor lymph nodes in tumor bearing animals, and we can show that we can regain this or expand these cells if we use our antagonists. So our experimental model right now is actually kind of twofold, but what we're thinking is that CD200 is secreted from the tumor site, and it actually acts on antigen-presenting cells, such as monocytes, mostly dendritic cells. And I'll go through this in a few minutes. What we've shown by RNA-seq analysis, that there's a nuclear factor, NFAT, that gets dephosphorylated and goes into the to produce TNF-alpha and IL-2 in the nucleus. However, we're showing, excuse me, that if we give the CD200 protein, we upregulate what's called PPIA. And this is also known as cyclophilin A. And what this does is it prevents or inhibits the dephosphorylation, inhibiting translocation, and inhibiting the cytokines needed for progression to an adoptive response. Moreover, PPIA downregulates MHC class 1s. So if we give it the antagonist, we're showing that we don't get upregulation of these genes or transcription of these genes. We don't get dephosphorylation as shown here of NF-AT. Let me see if I can show that a little bit better. You don't get the dephosphorylation of NF-AT, nor do you get the upregulation of PPIA. So our experiment model is that CD200 inhibits, expands, or upregulates PPIA, expands MDSCs, which results in immune suppression. However, if we use our CD200 antagonist, receptor antagonist, we're still working on if it's binding separate receptors or not, but we're hypothesizing it's going through the isoforms, inducing a response which induces chemotaxis, cytokines, chemokines, and ultimately immunogenicity. And I'll be showing you the data that led to this hypothesis. So earlier on, and this was actually published, we showed that if you use trans-well plates, and you put just 90 splenocytes on the bottom, and you put gliomas on the top with or without the antagonist, we can actually show that we get an expansion of myeloid-derived suppressor cell population. And with our antagonist, we actually get inhibition of this, or we don't get this expansion. And we just went ahead and showed it using known concentrations of the protein, plus or minus the antagonist, on normal naive splenocytes. And you can see here that you could, that you can inhibit the expansion of myeloid-derived suppressor cells, as well as arginase production, which leads to immune suppression. So we asked if this is happening in vivo. So we took glioma-bearing mice, and we vaccinated them with tumor lysates based off their glioma cells, with or without an antagonist. And what was interesting, and we repeat this a few times, is that you get this expansion of an MDSC population about day 28. And this is where the mice actually start dying. However, if we add our antagonist, you don't get this expansion. They actually go back down to the population percentage of a naive mouse. And we've showed this multiple ways, that in vivo, our tumor lysates, our gliomas, induce or expand a MDSC population, and we can actually now block that expansion using our antagonist. So being that we're focusing on antigen-presenting cells, we decided to look at the CD11Bs, which are monocytes. So we purified these out of splenocytes and did RNA-seq. We pulsed them with or without the antagonist. No tumor lysate, no ova, just the antagonist. And you can see here with this rainbow plot that you get multiple enhancement and suppression in multiple genes within the monocytes themselves. So we started asking questions. What are the effects of our CD200 receptor antagonist on bone marrow-derived dendritic cells? So we purified them from wild-type mice, and we just pulsed them with our antagonist alone, and we showed that we got beautiful production of IL-2, MIG, IL-1 alpha, TNF alpha, GM-CSF, IL-1 beta, and other cytokines. With the antagonist alone, we didn't use any kind of tumor lysate or ova, nor did we use an adjuvant. This is with our antagonist alone. We get a production of cytokines. Interestingly, we don't see that in monocytes. However, when you pulse them with our tumor lysate, plus our antagonist, we get this expansion or a synergenic expansion of multiple cytokines and chemokines. Moreover, if you look on the spleens, when you pulse with OVA, you get OVA plus our antagonist, you get this increased co-stimulatory expressions of CD80, 86, as well as MHC class 1. So we wanted to know if this is going to enhance our immunogenicity. So we went back to an OT1 mouse model, and we showed that if you pulse wild-type splenocytes with OVA plus poly-IC, you come in with these transgenic OT1s, which are already have the TCR for the syncytial, you get some kind of a response. But if you add our antagonist, you get a significant enhancement in immunogenicity. You don't get this if you use a scrambled CDR antagonist, CD200R antagonist. This is just our in-house nomenclature for it. Nor do you get it if you use CD200 receptor knockout animals. Moreover, on day 20 post-tumor inoculation, we harvested the splenocytes in wild-type or CD200 receptor knockout mice, treated with tumor lysate plus or minus our antagonist or our scrambled antagonist, and you can see that only in wild-type mice with our antagonist, you got a significant enhancement of a cytolytic response. In other words, we took the splenocytes, harvested them, incubated them with our gliomas, our GL261s, and we used this total cytotoxicity assay developed by ICT. And we showed that if you knock out the receptor, or if you use our scrambled, or you use just tumor lysate alone, you get a significant decrease in a cytolytic response. Now we're repeating this, but what was really interesting was we could actually differentiate apoptosis from necrosis, or cells that are that have undergone apoptosis are becoming necrotic, using their total cytotoxicity kit that they're developing. And this will allow us to tease apart mechanisms. So as I said, we're getting this enhancement of chemotaxis or chemokines. So we want to look at chemotaxis. Now, what we did to do that, to accomplish that, is we took animals with no tumors. These are just non-tumor bearing animals. And we treated them with our antagonist, saline, or tumor lysate alone. Either 1 or 24 hours later, we came back and we vaccinated them with saline for our saline group, tumor lysate plus an adjuvant for our tumor lysate group, or tumor lysate plus adjuvant plus our antagonist. 6 hours later, we went ahead and harvested the injection site. So now we're looking where we put the antagonist plus our adjuvants and tumor lysates. This actually is why we think that in the beginning, when I told you we have to use the peptide an hour early, this is why. What we saw here in saline is that if we looked in the capillaries and arteries, you virtually see just red blood cells. You don't see many lymphocytes. However, 1 hour or 24 hours later, you start seeing some infiltration of lymphocytes. We're phenotyping these now when you give the mice the tumor lysate. However, 1 hour with our antagonist, you see a significant increase of lymphocytes infiltrating in the tumor or in the vaccine site. 24 hours later, this is just chock full of leukocytes. So like I said, we're seeing some of them look like polymorphonuclear cells. We're phenotyping to see what's coming in. What we're hypothesizing is that this chemotaxis is allowing the infiltration of antigen-presenting cells, monocytes and dendritic cells, to enhance antigen uptake to elicit an immuno response. And you could quantitate this. If we took the 8 different sections of skin and just counted the cells that are infiltrating, we can show that our antagonists, I apologize again, it's supposed to be antagonists, significantly enhance chemotaxis or leukocyte infiltration into the area. And we repeated that, showing that if you knock out the receptor, you don't get that. So altogether, somehow I fixed these, somehow they didn't get saved. But altogether, we showed that using our antagonists, we could significantly suppress or decrease tumor burden, which, which significantly enhance survival of these animals. Moreover, we're showing that in our breast tumor models, we get almost an 80% survival of mice. And once these mice become tumor-free, we'll re-challenge them with up to 4 million EMT6 cells, but we don't have to vaccinate. None of the tumors will take post-vaccination. So, are there any questions right now? Because I'm going to switch gears a little bit. All right, so I'll go on. Now, we're capable of inducing an immune response now. We think we can significantly enhance immunogenicity using our CD200 receptor antagonists. But there's more problems to it. The tumor microenvironment itself is chock full of multiple suppressor cells, T regulatory cells, cytokines, IL-10, MDSCs, M2 cells, et cetera, et cetera. But what we found recently is there's this virtual force field around the tumor cells, which inhibit the infiltration of lymphocytes, we're hypothesizing. So remember before I told you that high-grade or these gliomas, GBMs, ependymoma, medulloblastomas have high transcription levels of the CD200. Well, when we looked further into that, we could see a brown in a normal tissue, CD200 staining, but that wasn't the case, as I showed you earlier, in glioblastoma multiforme. These are human samples. But what caught my eye with the help of a colleague of mine is this. We looked in the vasculature, and you can see here that normal brain within the endothelial cells of the blood vessels are basically have no CD200. You see the CD200 outside of it. That's not the case in high-grade GBMs. In high-grade GBMs, we get a lot of CD200 expression, upregulation of CD200 in the endothelial cells. Now remember, this is extremely immunosuppressive, and it interacts on the receptors, and the receptors, CD200 receptors, are upregulated on T cells. Moreover, this is tumor specific because if you look here, again, on a GBM, you can see the CD200, but you don't see it right next to the tumor itself. So this appears to be within the tumor itself. So we're thinking now that this CD200 inhibits infiltration or differentiation or differentiates cells into their suppressor properties. In other words, people have reported that it's this interaction between the tumor and the activated T cells, which bear the CD200 receptor. This on CD4, CD200 receptor positive cells differentiates into T regulatory cells, and on CD8s, they just render them inert. Well, we think that that's happening at this spot right here, that they don't actually get into the tumor microenvironment because of this surrounding immunological force field. Moreover, the CD4, CD200 receptors were hypothesizing. Now we're testing this hypothesis right now. We think they actually differentiate at this point, becoming T regulatory cells, and then they infiltrate into the microenvironment, inducing an immunosuppressive environment. And the same is true about tumor-associated macrophage. We hypothesize that these become MDSCs or M2 cells, and then they infiltrate into the tumor microenvironment. And this is where we're getting the infiltration of the suppressive properties, not by interacting on the tumor itself, because we're showing that the tumor itself virtually has very little CD200. So we wanted to test this theory, and also we wanted to know if we can overcome this. Now, as I said, CD200 receptor is upregulated on activated T cells. So I wanted to protect the receptor, so when they go back into this area, they're blocked and they can actually go into the tumor microenvironment without becoming suppressive or inactivated. Let me go back here. So what I did is I actually made antibody, a polyclonal antibody against the same epitope region as our antagonist, because we were afraid of coming in there with just an anti-CD200 receptor and inducing a suppressive response. So we made these antibodies against the specific regions of the CD200 as our antagonist. We inoculated tumors. Go ahead and do this. We inoculated tumors, and then we gave them the vaccines, which contained CD200 or tumor lysates plus the antagonist, tumor lysates plus a scrambled antagonist, or tumor lysates plus antagonist with the anti-CD200 receptor systemically on days 4, 6, and 13. And interestingly, what we saw is when we added the receptor with our antagonist, we actually got significant inhibition of tumor growth in that population. Moreover, by combining them, we could see that we had a significant enhancement in survival. Now, in this particular experiment, we didn't get overall survival, but we are actually giving the antagonist 30 minutes instead of 1 hour prior to vaccination. And I think that's why we didn't see a survival benefit with it alone. But by combining them, we did see a significant survival benefit. So now we're making monoclonal, and we're testing this with a monoclonal antibody. So we decided we wanted to translate this to a different model. So what we work with in collaboration with a collaborator of mine, Dr. Elizabeth Pluhar and Christopher Pinnell, is we derived a canine-specific CD200 receptor antibody. We take these dogs that come to the clinic, okay, we do not give them tumors. These dogs come to the clinic demonstrating disease, and we diagnose them with high-grade gliomas. We remove the gliomas, and we make vaccine, tumor lysate-based vaccine, and we added the antagonists to these vaccines with this set of dogs. And what we're showing is that the dogs really exhibit, some of them exhibit flu-like symptoms, which we've seen in our human patients who respond to immunotherapy. But what was really interesting is this is 1 out of 4 now that we're having a good response, is that when this is post debulking, you can see here that in green or in red now, that about 40% of the tumor itself was left behind. They were only capable of removing approximately 60% of the tumor. 4 months later, it's gone. The tumor's gone. Monday, this dog had his 8-month checkup, and the tumor's still gone. Moreover, interestingly, we see a lot of what we think is perivascular infiltration of lymphocytes into the tumor site, and we think that is inhibiting the ability of these tumors to come back. So we're comparing survival thus far. I didn't put it in this presentation because it's a little bit early, but what we're showing is that without treatment, glioma dogs live an average of about 50 days. With tumor lysate alone, we have a population of dogs that we've tested. About 214 days is the mean, and they drop off. There's about one dog out of 16 that's still alive. Well, some of our dogs are 248, 250 days out, and we have about an 80% survival on this ongoing clinical trial. So we're able to show in a murine that if you block this checkpoint blockade using an antagonist at the vaccine site, we can significantly enhance the induction of an anti-tumor response. Now, given that CD200s highly express on blood endothelial cells in the GBMs, and we've also shown this in melanoma and breast cancer, we think that we need to use a receptor antibody that'll specifically block the receptor without inhibiting the activation of the cells or downregulating the cells to allow it or to help it get into the tumor microenvironment itself. And that with this combination therapy, we'll be able to significantly enhance the immunogenicity of our immunotherapy programs against CNS tumors as well as in other tumors. So I'd like to acknowledge all the people from my lab and collaborators that have been helping me in this project. Thank you very much.
Thank you, Michael, for bringing us that informative presentation. Before we get to our audience questions, I just want to remind everybody watching how they can submit their questions. You can submit a question by typing it in the Q&A box, which can be found by clicking on the green Q&A button at the lower left. And we'll try to get to as many of your questions as possible. We'll just give our audience a minute to find the Q&A box and type in a question. That was an excellent presentation, Michael. Lots of really good information there. Okay. I think we have a shy audience today. I don't see any questions coming in. Michael, perhaps I could just turn it back over to you. Do you have any other final comments for our audience, what you'd like to take from, them to take from this presentation?
So basically, we're taking that there to enhance our therapy of children or tumor-bearing patients, our patients with diseases, rather it's CNS or other tumors, that we actually have a long way to go to discover that magical cure, and that there's not going to be one. I think it's going to be a combination of multiple checkpoint inhibitors at this point that will increase the efficacy of our immunotherapy. The PD-L1 or the PD-1 inhibitor is showing great promise, and that is addressing a suppressive response at a whole complete different site as our CD200 or CTLA-4. So it's going to be these combination immunotherapies or these combination checkpoint inhibitors that I feel we're really going to make a difference in the field. We have derived two candidate or two peptides that are human specific, and I didn't put that data in there, I could have, but we're showing great in vitro responses with them, as well as the same as we did in the murine. We're seeing a huge cytokine and chemokine production by taking immature dendritic cells derived by CD14 cells and just passing them with our CD200 receptor antagonists. So we actually have great promise that this will be translatable within a year or so, and hopefully we'll be in the clinic and make a difference.
So exciting, lots to do there. Good luck with all of that. I'd like to thank Dr. Michael Olin for spending time with us today and bringing us this information. I'd also like to thank today's sponsor, Immunochemistry Technologies, for making this webcast possible. I want to let our audience know that they'll be receiving an email from LabRoots alerting them when this webcast will be available for replay. We do keep it available on demand through May of 2016. So please forward that email to your colleagues who may have missed today's live event so they can get this information from Dr. Michael Olin. Thank you again for joining us. We'll see you next time. Goodbye.